A very good morning to all of you. My name is Shivangi Suman, and today I am going to anchor all of you through this program. So, first of all, I am really glad to welcome you all in this one day workshop on bioinformatics. Even if we are excited to bring together students, researchers, and professionals to explore this amazing intersection of biology and computational sciences. Since you are joining this workshop, I assume you understand at least what is bioinformatics. But even if you don't, I guarantee you will shortly understand at least the basics of bioinformatics by the end of this workshop. So let's start. So let's first discuss what we are going to discuss in this workshop. So we'll start with the introduction, then the biological databases, after that data mining techniques, and all the other points that we have mentioned in this workshop. I hope you can see that on your screen. That next we'll be dealing with homology search tools. After that, multiple sequence alignment, phylogenetic tree construction and analysis, protein domain and motif identification, protein structure prediction, so these are the topics that we will be discussing in detail in later on in this workshop. But before starting the actual workshop and before discussing all of these, let me first introduce you to the lab that has organized this complimentary workshop for all of you. So the workshop is organized by Flevin Labs Private Limited, and we are a tech-driven R&D based company. And the main motto of our laboratory is to convert, laboratory research into real-world products. We are an ISO certified laboratory, and we are registered in an MSME and Startup India. Along with this, we have received a grant from BIREC C Camp for one of our projects. Now, what are the works that we do at Fleming Labs? So, apart from research and development, we have certain products and services that we offer to the students and to other, for society and other professionals. But since most of our participants today are students, we would like to discuss some of the services that we provide specifically for the students to help them in their academic and in their professional growth. So the first thing that we offer to the students are summer and winter training programs, and these training programs consist basically from 15 days to 60 days. And if you want to know about the basics of basics in laboratory you want to learn, so this summer and winter training programs are really helpful in that. After this, we organize workshops of 1 or 2 days. So these workshops basically deal with specific topics or specific techniques that we could cover in 1 or 2 days. After that, we have 3 to 6 months trainings and projects. So these 3 to 6 months trainings and projects, the 6 months projects are basically taken up by MSc students. And this 3 months project, for example, there are certain BTech students and they have to do projects in their final year. So this 3 months projects are taken up by them. And even there are certain students who want to learn things in detail. They can learn things in detail in those 3 to 6 months projects. After that, we have dissertation and thesis. So there are certain MSc students who are working full time, and they don't have time to perform their laboratory experiments, or it's difficult for them to complete their thesis writing. So we help them in performing their things in wet labs, in laboratory, and then we help them in writing their thesis. One of the very important program that we offer is Biotech Finishing School program. So basically, Biotech Finishing School program is a 6 month program, and it is a guaranteed job program. We have partnered with certain industries because most of the industries don't prefer hiring freshers, and we have partnered with them, and we have taken the skills that they require in their employees, and we train students according to those required skills, and after that we help them in getting placed. So this program is very important for people who are in final year of their course, or they have recently graduated, or even they have graduated 2 or 3 years before. So and they haven't have job yet, so we help them in securing job. We also offer certain testing things. So we offer food and feed testing, we offer water testing, soil testing. Again, in academics, we offer dissertation and thesis for scholars and master students, and we also work in tissue culture. So we provide plants of banana and papaya plants, of tissue cultured plants of banana and papaya. And if you want to explore about our products and services, apart from these, you can visit our website. The website is mentioned here on your screen. Now, there are certain training modules that we provide. So, we provide training in microbiology, molecular biology, biotechnology, nanotech, mushroom cultivation, cordyceps mushroom cultivation. Again, cordyceps mushroom cultivation is a very booming market, and even Government of India is providing grant and is providing support for cordyceps mushroom cultivation. So, we provide training in cordyceps mushroom cultivation. We help students in learning those specific techniques that are really important for cultivation of cordyceps mushroom, because this is a wild variety of mushroom, and it's really difficult to cultivate this. So we provide training in cordyceps mushroom cultivation. After that, biochemistry, statistical analysis for life sciences. Again, this is a very important module for specifically for masters and PhD students, because once you have performed your experiments in wet lab, and now you have data, and you have you have to write your project, or you have to write your thesis. So you need to understand the statistics to make sense out of that data, and this module helps students in understanding statistical analysis. So this is very important for masters and PhD students. After that, linkage map and QTL identification, herbal product formulation. So nowadays, a lot of people wants to understand how herbal product formulation takes place. So if you want to learn a herbal product formulation either in pharmaceutical industry or cosmetics, or for nutraceutical formulations, we provide training in all of these. Now, if we talk about the time duration for which we run these modules for, so we provide um, both in short term and in long term. So, if you want to join the workshop for 1 to 2 days to 15 days, or you want long term modules, for example, from 1 month to 6 months, we provide two of it, and both of these modules are available in online as well as in offline mode. So you can learn with us both in online and in offline mode. So in offline mode, you'll have to join us in our laboratory that is present in Arjunganj, Lucknow. And if you want to do the online mode of certification, so we'll be providing you with experimental videos, PowerPoint presentations, study materials, and along with that, we'll be arranging live lectures for you, so all your queries can be dealt there, and we could teach you something new. Now, what you will receive if you do, join training with us. So you'll be provided with hard and soft copy of the certificate, and you will be given the welcome training kit. Welcome training kit is only for offline students, but along with the certificate, one of the very most important thing that you will get if you join the training with us is personalized letter of recommendation. So if you are you're applying for a job in any of a renowned institution, for example like CSIR, or you are applying for for the studies in foreign, so you need a letter of recommendation from someone from your industry. So we provide a personalized letter of recommendation to the students who have done training with us or have joined workshop with us. So along with the certificate, that is very important, we provide personalized letter of recommendation to our students also. Now the workshop that you're joining is absolutely free of cost. And why we are arranging this free of cost workshop, because the motto of Level Labs is to give back to the society. Since science has improved our life a lot, we really want to share this knowledge with people who are, and I think the best way to share this knowledge is to share with the students. So the workshop that we have organized is absolutely free of cost. So please join the workshop and be a part of the workshop till the end, so you get to learn what we are trying to teach you. But we are also providing certificates along with the workshop, and certification has certain charges, and these charges only cover the processing cost and the taxations that we have to pay if we generate certificates. So if you want a digital copy of the certificate, the price that you will have to pay is only rupees 99, and this rupees 99 is covering the taxation charges and the processing fee that we have to pay when we generate certificates. For example, we are generating 500 certificates, so we'll have to pay certain amount of taxations over generation of these certificates. That is why we are charging this minimal amount of rupees 99. And on the special request of some students, we are also providing hard copy of the certificate, because sometimes a soft copy of the certificate get lost, or if you're presenting yourself in an interview, it's more credible to present hard copy of the certificate than to present the soft copy of the certificate. So we are providing hard copy of certificate in rupees 499, and rupees 499 cover all the charges, that is the printing charge of the certificate, the lamination charge. Along with this, we'll have to pay the processing cost and the taxation, and this hard copy will be dispatched to your location. So the cost of dispatch is also included in this. And keeping in mind that most of our participants are students, we are providing a coupon code of rupees 500, and you can, along with the hard copy of the certificate. So whenever, if you take any of our programs either in online mode or in offline mode, then you can redeem this rupees 500 rupees coupon code. So I don't think it's a bad deal, because you are taking certificate from rupees 499 and we are giving you back rupees 500 coupon code. So you can redeem it whenever you are joining any of our training programs. Now, why it is worth to take the certificate? First of all, certificates absolutely enhance your CV. So for even if you have learned something, but you need a certificate to prove that you have learned something, that. Along with this, why it's good to take certificate from us, because as we have discussed earlier, we are an ISO certified laboratory, and we are working in this field from a long time. um, Along with this, let me show you the certificate. So this is our certificate that you will get, hard copy of the certificate. So in the top left corner, there is a serial number given here, and this serial number will be fed in our database. So suppose you apply for a job and your employer wants to check the credibility of the certificate. So at the bottom here, our email ID is mentioned. So your employer can mail us, and we will be uh, to cross check ki is the certificate credible, or if this person has attended the workshop or not, and we'll be approving that yes, you have been the participant with us and you have taken the certificate from us. So your certificate will get a cross check also. This will also provide credibility to your certificate. So this certificate will absolutely help you with enhancing your portfolio and your CV. So please consider it if you want to take. Now, if you want to take the certificate, we'll be sharing a QR code and payment link in the group. So you can make the payment from there, take the screenshot, and then there will be a Google form shared. Fill the Google form, and we will be providing you the certificate within 7 to 10 days of working days. Now, the chief speaker and the lecturer of today's program is Mr. Sandeep Kumar. He is R&D head of Levin Labs Private Limited. So he will be taking your session today. After that, Mr. Kuldeep Mishra, he is the senior research associate and he is the program coordinator. So he has coordinated all the programs. And there goes I, Shivangi Suman, junior research associate, and I am anchoring you through this program. So thank you so much, all of you. And now let's get into the details of the workshop. I hope that you'll enjoy the workshop. Please be the part of the workshop till the end. And please keep posting your queries that you have to ask while going through the program. And at the end of the session, we'll be answering all the questions of yours, because it's really difficult to keep on, if you'll keep on asking questions, we cannot answer every question in the middle of the session. So once the session ends, we'll be answering, all the questions. So keep learning. And now I would like to invite Sir to come up and start the presentation. So thank you. Happy learning.
Uh, Thank you, Shivangi. Good morning to everyone. So, let's start with the workshop. Okay, so my name is Sandeep Kumar, and I am Agri Biotech R&D Head of Flamin Labs. Today, I am taking this lecture for all of you. The title, as you can see from title, it is From Sequences to Insights. Okay, so we will be dealing with whatever sequences we have, and we will try to decode it and get into a certain result. Okay, so these are the content which we are going to uh, cover today. So first we will take a brief introduction, then we will shift to databases, and from databases we will get to data mining, and from data mining, and we further proceed on the processing of that data, whatever we have collected. That is through homology search, multiple sequence alignment. Then we will shift to phylogenetic analysis, and in last two topics will be related to protein. That the first one is domain and motif identification, and next one is structure prediction. Okay, so we will start with bioinformatics. What is bioinformatics? As its name suggests, it is an amalgamation of two terms, biotechnology and informatics. So it is a integration of biology, computer science, and information technology to analyze and biological data. So, and what are the key goals for it? It is for retrieval, analysis, data storage, and prediction of whatever biological data we have in our system. uh, you must ask a question whenever you are learning something new. So first question should arrive that why bioinformatics, and or we can say what is the need of bioinformatics, because we are currently living in data driven society, and every minute, every second we are generating a lots and lots of data. So in manual track of this data and manual analysis of this data is near to impossible thing. So, with help of databases and advanced computer technology, this data analysis have been very easy with help of uh, bioinformatics. Second is acceleration discovery. As we all have gone through this COVID, so at that time, you must have seen that vaccine development and test kits developed very fast. So bioinformatics plays a major role in that. If you look back into the history, and you can see that pneumonic plague or any other disease, whatever had occurred, it took a long of time to get the cure or even identify the main causative vector. With help of bioinformatics, we have tackled the COVID disease in 2 years. Next one is bioinformatics help in enabling personalized medicine. As we all know, each person, sorry, each person is built up differently. So with the help of biotechnology and bioinformatics, we can personalize the medicine and get it person cured very fastly. And last but not least is bioinformatics bridges gap between different disciplines of biology or life sciences. So next topic is databases. Whenever we talk about databases, we always think about something related to computer science. But in reality, database is a collection of a large data which is designed to store, manage, retrieval, or analyzed on the command. Okay, so whatever data we have, we are keeping it as a single place. We can call it database. Database can be made for DNA, can be made for proteins, can be made for any literature search, or can be made for any protocol. For example, if you are working in a laboratory and you have made a folder which contains all the protocols, whatever you are going to do, you can save it and you can call it local protocol or manual database or SOP database. it is used for efficiency, that whenever you have everything labeled, well labeled, or everything well stored, then you can retrieve it very fast. It supports research whenever it is in global platform in cloud, then anyone can access it from any place of the world. Okay. Plus, daily new researchers have been adding a new data in these already established databases that which help other researchers to perform their work very efficiently. So there are mostly three types of database. First one is primary database. Primary database are the databases which store data as in raw form. There is no processing, there is minimal labeling, and there is no analysis that has been done. If you want to study a data, sorry, if you want to do a new research, but you already know that some person have done data deposition already in databases, you can just get data from there, and you can do your own start from the scratch. Only thing you will be avoiding that you don't have to go for sequencing or whatever technique used for database generation. Okay, so few examples of the database are like GenBank NCBI. That is a vast laboratory of library of databases, multiple databases. Then EMBL EBI is it maintained by European Molecular Biology database. DDBJ is another database which is maintained by Japan, and PDB is for protein. What are the key features for the primary databases? It is publicly available. Researchers may submit new data or can update it also. But the main point is that it is a raw entries. If you want to correlate it, you can correlate it like you are going to sabji mandi or market and getting your all raw ingredients from there. From raw ingredients, you can make anything and any dish you can whatever you desire. So if you correlate with in that term, so the primary data is our uh, main big market. Okay, then we will move to secondary database. It is slightly more curated and more processed database. It contains it takes data from primary database, do the analysis, interpretation, annotations, and then store it in processed form. Okay, so examples for this is UniProt KB. It contains annotated protein databases, all the protein sequences which are already annotated. Similarly, PFAM. PFAM is a collection of protein families. Interpro, and you can from Interpro you can do the integrated database. It is for predictive models. What are the key features? Key features are that already data it is in process form, so it have been validated, it have been grouped and interpreted by the experts, so there is a less chance that you will miss anything or misinterpret anything if you are a novice. Okay, so third type of database are specialized database, that is niche focus. They are limited to a single thing. Mostly your miRNA database or your pathways and your transcription factors, these type of things get into specialized database. Examples for these are CAG, Kyoto Encyclopedia of Genes and Genomes, MIR base, HGMD, plant TVF. So these are very highly specific, provides only highly specific information, and they cater the need of a very specialized research committees. Like if you want to work in this microRNA platform, so you will go to my miRNA base to get your sequences or information whatever you need. You need not to go on the primary database like NCBI to get information of miRNA, because that is raw data. Here you will get specific and catered data for yourself. Okay. So just I have provided a summary so that you can get a gist of whatever we have discussed till now. So primary, secondary, and specialized, these are the three types of database. Primary contains your raw experimental data. Secondary will your curated and analyzed. and specialized will focus on a specific domain. Okay, examples: gene bank and PDB are examples of primary database. UniProt PFAMs for secondary, CAC microRNA databases for specialized. And here I have mentioned the analogy like primary, you can consider it as a warehouse of ingredients. Second is ready to cook meal that already someone have curated and assembled everything. Just you have to put some little efforts and you will get whatever you need. And specialized is like a specialty store that are specific to single thing that whenever you need, you don't need to search for in warehouse. You will just go to directly specialty store and get your things done. So we will move to next topic, which is data mining. So data mining is extraction of your useful patterns from large database. For basic bioinformatics, and mostly if you are new to bioinformatics, whenever you hear the term data mining, first thing should come in your mind is NCBI, because NCBI works as a database as well as search engine for all the data. So we will do a short exercise where we will see how to mine data from NCBI. Okay. So NCBI's domain is ncbi.nml.nih.gov. It is maintained from USA. You can simply search in Google that NCBI. Whatever first hit you will get, it is lead to you this page. If you want, you can we open a Windows side by side and do check whatever I am telling you. Okay, so if you see this page, uh, you can just check that if I click at all database, you will get a number of things like assembly, bioproject, biosamples, gene, genome, and if you go down, you will get PMC, protein, and PubChem, PubMed. PubMed is a database for your all scientific literature. Whatever research article have been published and is open access, is always available with PubMed. If you want to download any paper or read any paper, you can just simply go to PubMed. You can just search just like Google, and you will get your papers from there. Okay, so today we are going to do exercise on the base of nucleotide, and so we will first let me just give you overview of page here. As you can see on your left hand, you will get all the resources, that is chemical, bioassays, DNA, whatever resources are there mentioned is there. Your popular resources, in resources, you can say it is the database. Popular database are listed on your right hand side. On home page, you can see six different tabs that submit, download, learn, develop, analyze, and research. For submit, if you want, if you have your own data from your own research, you can submit it. So you can click on that and it will guide you everything. You can transfer all the NCBI's data to your local system, because if you are going to do any core bioinformatics analysis, that is just, you cannot rely on the internet speed. You cannot wait for, because on the server it will get uploaded, then processed, then it will show you results. So it will take a little time. Plus sometimes when you are dealing with large data set, you can't upload it like I'm talking like large database like 5 uh, GB, 10 GB, or 1 TB, then you can't do. So you can NCBI provided a facility to that you can run all the programs on a stand-alone basis. You can find the data documents on NCBI which contains the classes and tutorial for whatever is stored in NCBI. Any tool which is driven by NCBI's list, you can learn for it by watching tutorials or by reading. So either you can go to YouTube. Instead of YouTube, you will get in YouTube, some other person will explain it to you. It might be helpful, might not be helpful for you, but whenever you are using from this NCBI's database. So they provide a whole list from scratch, from zero to result interpretation. You will get from here. And so let's not waste much time on this. We will directly jump to our exercise. We will open nucleotide, and you can search any nucleotide from here. For example, I want to download actin gene for anything. So you can, it is not case sensitive. You can type any way you like actin. Okay, you can click on search. It will take like 2-3 seconds, and you will get all the sequences or all the information from the nucleotide database where have been actin is mentioned. Okay, so as you can see, uh, it is showing like 88. Sorry, I misspelled actin. Okay, so you can see it is telling that it have found total much 6,72,405 sequences. Okay, so now you have to filter out to get your data. So on your left hand, you can see you can filter it by species. That if you want to work on animals, and then you select animal, and it will show only results for animals. Same for plant, fungi, and all. You can filter your search result on the basis of molecular types. You can select your database. You can select sequence type. Whatever you want to get selected, you can do from there. For this workshop, we are keeping it very simple. So we will take first result, and we will proceed with that only. So this is the summary page for our result. So it is telling what it is telling. It is telling us that we have selected the sequence of gallus actin, and it is a beta actin, and it is a mRNA sequence, whatever we are viewing here. It will provide a locus ID, and it will provide a reference sequence, and this is its SSN number. SSN number is unique for each and every sequence whatever you deposited in the NCBI. You can correlate it like your Aadhaar number. As we know that Aadhaar number, your personal identity number, is attached to a single person. It shouldn't and it won't repeat with anyone. So here, if this is a NCBI reference sequence or SSN number for this particular gallus actin beta actin gene. Okay, so and here you can see, as with the SSN number, it is giving a version, like at the end it is mentioning that point 2. What point 2 is denoting? Point 2 is denoting that researcher must have submitted it earlier, but he on the course of work and course of time, he have added a new information in these sequences. From here, you can get the detail of organisms, whatever organism and whatever its classification list is there, who have been submitted. If it is been submitted and used in any research article, it should be mentioned here. From which journal, what is the PubMed ID. If you click on this link, it will directly lead to you the paper research article wherever it is mentioned. Okay, so next we will scroll down further down, and we can see that here in CDS sequences, it is giving us information of about exons. It will provide you information of CDS. CDS is coding sequences, so it will directly convert into protein. So you can see that there is a mention of protein ID here. If you click on that, it will lead to you directly protein for this particular sequence. Okay, then translation, nucleotide translation to protein is also available there. If it is complete CDS, and it is by complete CDS, I mean it should contain a star to stop codon. If it is complete CDS, it will always have a protein translation with it. And if you further scroll down, you will get the sequences. This is the sequence of our gallus actin gene, sorry, or nucleotide sequence. As you can see, it is mentioned in ATGC format. this single chunk is of 60 base pair, so it is arranged in chunk of 60. Now we have to download this sequence because you can't use in this numbered format, because if you copy it, as you can see, if I'm copying it, it is selecting all the numbers also. So we have to go through the hassle of removing all the numbers, then arrange it. Plus, it is not very representable. So, how to download it? You can just simply go to here, top, in your right hand corner. You will see the send to option. Okay, from send to, you can click on coding sequences, and you can click from here, choose that you want faster nucleotide or faster protein, or if you want complete sequence, then you can click on complete sequence, go to file, and select your format. And whenever we are downloading a sequence or using sequence, we will further using, we always prefer to download it in FASTA format. Okay, keep always this in mind that you have to download sequences in FASTA format. You click on this and then create file, and it will directly start downloading it. Okay, so what is the FASTA format? If we You can see from here, if you click on this FASTA, it will lead to you another page where you can see this is you must be familiar with this format. This is called FASTA format. It is a standard format to store any nucleotide, any protein data. Because as we know that formats are instruction to the your laptop or your computer, that this use you have to read this file in this way. Like if you have seen that you store any word file, Microsoft Office word file, it will always store in dot word. If you are doing Excel, then it will store in dot xls or Excel format. Like images, in case of images, it will store it as dot jpg format or dot pg or dot png. So how to identify that whether our data is stored in FASTA format or not? So you will always see a greater than symbol at the start of your sequence. Then it will contain something, some text, whichever its SSN number, it's what is the sequence denotes, and type of. The first line is your always stored is like this, and this first line is called as identifier line. It provide softwares to identify from where we have to consider any string as a sequence, because as we know that our genomic data or nucleotide data is always stored in ATGC format, and this alphabet get repeats in our names also, our identifiers also. So this is the way to discriminate from whatever is our introduction or identifier line, what from where our sequences is started. So first line will be always identifier line, and then there will be a line change option, and then you will get your sequences. So I guess you all must have learn that how to download a sequence or how to retrieve a sequence from database. Similarly, you can download protein information about your RNAs, mRNAs, or microRNAs. Similarly, there is a slight variation in each different software, but basic principle is the same. Okay, so okay, so we have completed one system, that data mining. One thing I want to remind you all that please, if you are getting any net problem or your system is video is getting buffered, don't worry. This lecture is being recorded, and you can watch it later. Okay. And if you have any question or any queries regarding anything, whatever I'm telling, you can paste in your chat box. And at the end of the session, we will take the questions, and I will try to solve those queries as good as I can. So next one is once we have downloaded data, Okay, so now we have to analyze it. That we have to go and see what it is saying. Okay, so first thing is that whenever you get a data, you always go for verification of it. It is a simple human tendency that if we are getting anything, we always verify it. Like first is day-to-day basis example is whenever any one person is handing you a 500 rupees note, it is in our inbuilt habit that we always either try to feel its thickness or pick it up and watch it for holograms and all everything. So whenever you are plus homology search is a very good tool for your identification if your sequences is unknown. For example, if you are working in microbiology lab, you have developed we have identified a novel microbe for your any particular purpose. Okay, so now you can verify it with help of this morphology, it's biochemical. You can cross refer to literature and say that if colony shape is like this, colony size is like this, and it is this test positive or this test negative, so it must be this particular bacteria or fungus or anything. Okay, so but the question still arises that when we are going for strain identification, you can't tell that just on the basis of its morphology and its biochemical test. There you get its sequence, amplicon sequencing. That is the term for sequencing. It's amplicon, the 16S sequencing. And so from there you will get a nucleotide sequence. Okay, so how to identify or prove that whatever you get as whatever you are claiming. So homology search is the tool for that. Okay, homology search, what it does, it look for its similar sequences in data, all databases already someone have provided. It aligns it, like it superimpose it, as you can see the image that I have tried to show, that it is similar region is get identified of the two different species. Okay, so it's whatever it is similar, it will get aligned, it will get superimposed, and you will get a certain result. Okay, so what is the need or what is the purpose? So you can do similarity search, that sequence alignment or database search. You can, if its sequence is similar, it's so l- logically its function should be very similar, so we can do functional annotation. If its sequence is similar and everything is aligning, then structure should be always similar. Mostly theoretically it should be similar. And plus you can homology search, from homology search you can lead to phylogenetic analysis, evolutionary analysis. Okay, so homology search, BLAST, that is basic local alignment search tool, is the most widely used tool for homology search. Okay, so it identifies the local similarities within the sequence and give you the result on the basis of how good or how bad it is aligning. Okay, so here I have tried to provide a single workflow that first you will get your sequence, you will input it, then it will search in database and see with which sequence it is matching best. Okay, after match, it will uh, go through different algorithms and provide you search results. Okay, so whenever you must have heard about BLAST in your coursework or in work of your research. BLAST is a very basic and very widely used tool. So there are mostly four types of BLAST. First one is nucleotide BLAST. As its name suggests, it will take nucleotide as a query, and it will search in nucleotide database only. Then second most widely used is protein blast. Protein blast is for protein to protein alignment. That for it means that it will take protein as your query, and then in protein with database it will search and try to align. There are two main subtypes, that is BLASTX and T-BLASTN. In BLASTX, you will provide nucleotide as a query, but it will give you result and in terms of protein. That is, it is queries your nucleotide. it will convert into protein, then search in protein database and give you result in terms of protein. Okay, and then T-blastn is opposite of that. It is take as protein, it will reverse transcribe it, it will convert into nucleotide, then go to nucleotide database, search homology, search for there, and generates results. Okay, so this is the basic. I have provided a simple flow chart that what you have to do is if you are doing nucleotide database or BLASTn. First, you will go to BLAST website that is provided. You can simply just Google it, BLAST. It will give you there. You then you will paste your sequences. Then you can filter out that whatever specific filters you want to take, like if you want to blast this again any organism or number of results, whatever parameters you want to change. These are optional parameters. If you are novice, then you need not to change in that. Then you will click on next, that is nucleotide blast. Then you can choose databases also there, and then click blast. Okay, so we will do a quick uh, exercise for that also. For example, that I have taken gallus from here, and what we will do, we'll copy it. We'll go for NCBI. Okay, sorry, just I've already completed because these exercises takes time because these are running on server. So I have just already done so that we can quickly shift to there. Okay, so this is the basic. If you search for blast, this will get page like this. Here you can see that you can select nucleotide blast. Okay, so this is the home page for your blast n. Okay, so this is place to provide your query. Query can be submitted in form of different forms, that you can provide SSN number only, you can provide GI, or you can provide faster sequence. If you are doing this exercise with known sequences, then you can provide an SSN number also. If your sequence is unknown, you have sequenced it and you got from there, you can just paste your for sequence in faster format. One thing you have to always keep in mind, that you have to submit sequence in fast. If you are submitting a sequence, then it should be in faster format, that your identifier line should be there, else it will not give you a proper result. Okay, then you shift down, you can see there is option of databases, that you can see that is standard database. You want to blast it again, rRNA or ITS. If you are working with microbes and you are working for amplicon sequencing, then you can directly select rRNA or ITS database. Then genomic, or if you are since pandemic we were working hardly on beta coronavirus, so they have provided that shortcut for there also. Then you can select any database whatever is you are required from here. But usually if you are novice and you don't have to need much filter, so we select only core nucleotide database. Okay, you can exclude or include any particular organism. If you type a name of like if I do the mangi Vera indica. Okay, for example, what it will do now, it will search in database wherever Mangifera indica is have been cited, and it will I'll try to align with Mangifera indica only. Okay, you can add multiple organisms by clicking add organism, or you can say that if you don't want Mangifera indica, you can select on exclude. It will not search wherever Mangifera indica or Mangifera have been cited. Okay, so we are keeping it simple, and we won't change anything, and we will just we you can select that how much stringent your condition should be. By stringent means all the parameters, all the algorithms should be relaxed, or should be focused on getting you best result. So So you can select highly similar sequences, more dissimilar sequences, somewhat similar sequences from here. Okay, then you will simply click on blast or select show a result in your window, then click in blast. Okay, so it will take little time, as you can see, but I've already prepared result for you so that we don't get waste on time. So here you, it is a result of blast. As you can see, it will provide a job title from here. It will provide whatever program you are doing. It will provide a ID. You can always refer back to this ID and get your result. If you are logged in the, if you have just like your login, you can log in with your Google account or whatever email ID you want to share with them. You can log in, and once you logged in, this RID will be saved in result section, and you can identify from there only just if you want later. Okay, so how to interpret the result of this blast, because you have done blast, now you will get this this data sheet, you will get graphical summaries, you will get alignments, and you will get taxonomies. But how to interpret it is the main thing, because if you don't know interpretation, you can't correlate it, then it is of no use. So whenever we are interpreting the blast result, we always look for three things. First one is e-value, second one is query coverage, third one is percent identity. Because in the, if you can see, there are almost 100 sequences. It have given back 100 sequences. You can't decide which one you should consider. For example, if this one is a novel sequence, okay, how to tell that it is whether it is beta actin or it is something else. If you go, like for example, in terms of microbes, if you are doing applicon sequencing, then which one you should prefer. So we always go for first e-value. E-value should be 0. If it is 0, that means it is aligning 100% best match. Okay, it should be either 0 or in negative forms. It shouldn't be positive, because positive value is very bad result. So always look for e value. Either it should be 0 or it should be negative. Then we will go for percent identity and query coverage. Query coverage identifies how much length it have covered, how much your query have been aligned with your test subject. Okay, so here 100% means from start to end everything have been aligned. Percent identity again shows that how much percent your alignment have matched. So for this we can easily get to understand from alignment page. As you can see here, it is giving it alignment result. Okay, so you can see our whole sequence have been aligned perfectly with our query. So this percentage made means percent here, percent directly means everything is aligning perfectly. So this was meant from here, percent identity. Now you can, if you are submitting a sequence or you are proving that, if I'm saying that this is beta actin, then you can just take a screenshot of there. You can prepare a table from here, or you can just simply click on this graphical summary and take a screenshot of this data and provide wherever you have to prove that. If you are saying it is gene A, then it is gene A from here. Here it is, you can see that it is color coded. If it is in red color, that means alignment score have been is either 200 or greater than 200. So it is very perfectly aligning or very good. You're getting maximum query coverage, maximum everything. So we'll get uh, alignment from there. Okay, you can just simply um, if you want to make a phylogenetic tree, or you want to do any exercise which is related from here, you can simply download from here only. You need not to go further again on blast and re-download is everything. You can see select all, or you can first deselect and select whichever are applicable for you. So you can just, like for example, I have selected a few sequences, 5 sequences from here. I can go to download. I can download complete sequences of it or aligned sequence. Complete sequence means whatever percent is not aligned. Like for example, here if you can see, In second, it is aligning only 88.95%. So if you click on download and align sequence, it will download only 88.95% of this sequence. It will remove any unaligned sequence. If you click on aligned sequence or complete sequence, it will download 100% of the sequence. For example, I need complete sequence, so I will click on first step, and then it will prepare a new file, and it contains all the five whatever we have selected. Okay, hope I am not rushing, and you will all be able to understand it is quite clearly. So this was the our homology search. Okay, now we will move to next segment, which is multiple sequence alignment. As we have seen in BLAST, BLAST is was local alignment search tool, and it is aligning only two sequences. One was our query, another will be your subject from the database. It is aligning against different sequences, but at a time it is showing result for two sequences only. If you want to analysis of do the analysis of two or more sequences, you have to go for multiple sequence alignment. Multiple sequence alignment can be done for DNA, RNA, or proteins. Okay, so what is the purpose? Its purpose is to get understanding about an evolutionary analysis. You can do functional prediction. You can do structures, look for a structural insight, and you can do study about phylogenetics. Its application is comparative genomics based. Comparative genomics is wholly based on multiple sequence alignment. You can do the motif identification, or if you want to classify certain proteins, if you are working with proteins, then you can go for multiple alignment. Okay, so for multiple alignment, Clustal Omega, it is a widely used basic tool for MSA. Clustal Omega, what is it will do? It will take your sequence. It will if the one main difference between multiple sequence alignment and BLAST is that in BLAST you get sequence from the database, but in case of multiple sequence alignment or MSA, you have to provide all the sequences whatever you want to get aligned. You won't get any sequence from the database automatically. So first you have to need your 2, 3, or 5 or 10 or whatever you want to do. You have to make sure that you have data. If you don't have multiple sequences, then you can't do multiple sequence alignment for them. Okay, so uh, so what are the key features? This is the progressive alignment that it utilize a guide tree to align sequence in a stepwise manner. It have HMM profiles, that is hidden Markov model. It used for enhancing accuracy. It can be scalable, that it can handle 2, 3, 5, 10, 500, or many data at a time. Its input and output, its input format, as I have already explained you, that input format is always in FASTA format. You will get output in multiple format, like Clustal format, FASTA format, Phylip format, is got Stockholm format, any format whichever depends on what tool you are using. Okay, so what is the its workflow? It's simple, that you open the Clustal Omega, its web interface. You will paste your input sequences. You will check any changes. If you want to make any changes from the basic default profile, you click on alignment and you will get the result. Okay, so I have already done. So this is the first. We will open the cluster. This is the home page for Clustal. Simply again, you can just Google Clustal Omega. You will get the result for the Clustal Omega website. It's EBI. There are two types of Clustal you will get if you search only Clustal. One is from DDBJ and second is EBI. So depending upon whatever your need is, but if you search for Clustal Omega, it will always lead to here. Okay, so you will paste your sequences here. You can select either do you want to DNA or do you want to align RNA. in our case, we were working with our nucleotide sequence, so we did with the alignment. Then in parameter form, or if you want to see, just you can click on. If you are novice, then always know that there is a always this example file is always present in every test, so you can use that just to get understanding about that. You can choose parameters like if you want result in with character counts or without character counts, so you can choose from here, or you can see that multiple formats and everything is there. So it's just keeping it basic. We are doing with Clustal W. Then you can submit it as a title. Title is required whenever you are dealing with large data set and multiple MSAs at a time. So you can just put a name here. You can do whatever in terms of alphanumeric terms, and you get submit. And it will take a lot's time, because in case of blast you was doing one-to-one search. Here you are aligning multiple sequences, so it will take little time. So it will generate a ID that you can later refer back. I have already done all that. So here you can see, once you are done, click aligned, then you will get an alignment result in this format. Okay. Here is this job ID. This job ID will be valid for 7 days. On the Depending upon load on their server, sometimes it get valid for 3 days, sometimes it get up to 7 days. You can copy it and keep it with you if you want to refer back later, but within 7 days, not more than 7 days. So this is the alignment result. How to decode this? As you can see, this one are the our that what to say. Uh, SSN numbers. Here you can see it is mentioning number at the end of this number denotes how much sequence have been aligned or how bit sequence has been used for alignment. For example, if you can see that for numbers of sequences it is saying 0. That means it is have 0. It aligns sequence in a way that best alignment should be there. So it provides certain gaps in between sequences so that it will get best match. As you can see here, lots of gaps are present. That means our alignment have been not done yet, and we haven't find any constant uh, domain or constant preserved sequence. Here you can see we have got lots of T, but these T's are consistent over all the sequences. And similarly, this stretch is consistent. So whatever is matching perfectly, aligning perfectly, you will always get a star below that. If it is not aligning, then you will get a blank. This blank is called gap. On the base of algorithm, it will generate this gap to get best alignment. As you scroll down further, you can see more and more sequence are getting aligned. Okay. So how to use this in case you are dealing with certain you want to prove that no, my sequences or my this microbe is unique, then you will always look for these gaps that your sequence should have must have it different from the already submitted in database. So if you identify this gap in your particular sequence, then you can say, see it, this particular number, like you can get number from here. At this number my sequence have A instead of G or T or whatever it is, then you can say that this particular sequence is unique to my particular organism. That's why I am saying this is strain, a different strain of this. Okay. So from here, and you can just scroll down and see that it is widely distributed between matched and non-matched sequences. Okay. So you can generate a phylogenetic, you can color code it also. If you click on color, it will color code on the basis of nucleotides. Then you can see, and if you go scroll up, then you can generate a phylogenetic tree from here. This phylogenetic tree, you will get a phylogenetic code here, and then because a very long sequence, so it will take a little time to generate a phylogenetic, but it is a very raw type of phylogenetic tree, and you will get just a basic idea about your alignment and ancestry. So you cannot do much alteration here. So we will be doing this phylogenetic analysis with different softwares. So this covers our MSA. From MSA, now we will move to phylogenetic analysis. That once we are done with MSA, we have already identified that our sequence is unique and this one is different. How to represent that in presentable manner? Because you can't use this kind of phylogenetic tree in papers or for good papers. You can present it for in front of your mentor or in front of your guide, but it can be it is not very eye pleasing. So we will go for phylogenetic analysis. So what is the phylogenetic analysis? It is a study of evolutionary relationship among species or genes based on similarity and dissimilarities on their genetic materials. Okay, you need a sequence, you have sequence, you have retrieved sequence, you have retrieved similar sequence to that. Now you want to do that analysis that whether our sequence is similar or non-similar. You done that with help of MSA. Now you want to uh, understand and present in better way. So phylogenetic analysis is done for that. What are the purpose? First, you can always trace its lineage. You can study about evolution. You can identify its ancestors from where it came. Okay, so phylogenetic tree. First, we will understand about phylogenetic tree itself. That what it represents, what it is saying. Then we will do the exercise, because if you don't know what it is saying, then it is of no use in making a phylogenetic tree. It's just a cosmetic purpose that you want to show it's aesthetically pleasing. That's it. So from phylogenetic analysis, from phylogenetic tree, from where that your this is starting, it's called as a root. Okay, sorry, then it will diverge and form a just take it inspiration from the tree or our family's tree. As you know that tree, it contains a root. From root it will get, you get a stem, then it will further and branch will divide its primary branch, secondary branch. Similarly in phylogenetic tree you will get a root, then it will diverge into branches. Okay, and whenever branch is further divided into sub branches, that point is called as node. Okay, at the end points, whatever the end points are there is always referred as a node. And when uh, two or three or more branches are coming under same node, they are labeled as clade. Okay, so as I have represented here, that you can see that it is starting from a root, then you get two branches, then our topmost branch get further bifurcated, and then again bifurcated. So this species A and species B section, if you can see that they are coming under single node from here. So prime single, if we ignore later on section, you can say this is a common root. Then from common root, they are diverging into two. So these two species A and B will be always considered as clade. Okay. So plus this length, how much distance it have been from the root, is directly corresponds to its genetic trait. Like if you see that species F, if you consider species F and species A, so you can see that species F is very close to root, but species A have been bifurcated twice. Okay, so that means species A and species F are very uh, dissimilar in terms of genetic makeup, while species A and species B have been very close, and so you can say that these are closer ones. Similarly, you can interpret the result. So there are basically two types of phylogenetic tree. One is rooted, second one is unrooted. Okay, so rooted tree, that you can see, as I have shown previously, that you will start with a definite start point. Clear hierarchy is present. A designated root is always there. So it is called as rooted tree. But in case of there is no common ancestry, no direct evolution is present, so it is always like in second example, you can't say that from where it started. Everything is have been bifurcated, everything have been separated, but we don't have any clear endpoint. So this is called as unrooted tree. Okay, so it doesn't have always don't have any common ancestry. There is no direction of evolution. No direction of evolution. By this means, if you see that rooted tree, you can say that this whole tree, that A B have been derived from the common root from the C. So, but here you can see that that they directly evolved from this or that. It can be from here. It can be from here. Okay, so these are the rotating. So phylogeny.fr is a web-based tool. It is a very basic tool and very easy tool to perform this phylogenetic analysis. It is very beginner friendly. You don't have to tackle with the lots of format and everything. So first we will do an exercise with phylogeny FR. Just simply search Google for phylogeny FR. Okay. Whatever first time is here, you select on that. Let it open. I've already downloaded some sequences, so we'll using it. This is a homepage phylogeny FR. Okay, so citation how to cite it is always there. We will just do. We will go to phylogeny analysis and we will simply select one click. If you don't have knowledge of much about how to make everything, one click is big board and it is very good for your primary analysis. You can paste your sequences here. Always remember it takes your file in FASTA, EMBL or Nexus format only. So anyone can be used. You just click submit and it will take like 1 to 2 minutes and it will give you a phylogenetic tree. So see here it is in move to next step. It is now aligning the sequences, aligning on the basis of MSA or cluster. Whatever we have done, it is doing so. Now it is time to generate your phylogenetic tree on the basis of whatever algorithm it have been already fed. Yeah. So as you can see, it is generating tree on the basis of maximum livelihood algorithm, and now it have completed identify, and we will get a phylogenetic tree from here. You can see that it is very um, compact tree. You can change everything on your requirement. You can select tree style, that is this phylogram you want, cladogram you want, radial tree or circular tree, whatever you want. You can just select from there. Okay, you can change, and you can download it is in format of PNG, PDF, SVG or any format you want. Here you can see that we have provided number of sequences. It was acting for different type of plants. Okay, so here you can see that this one is start of the point, and it get first divided into two main things. That first one is here, other second whole group is there. This that Arachis hypogea and Cagnus Kajan is singly they are coming one side, and dust dollar they are going on the other side. In here you can change and colorize leaf, plant size, whatever you want, but still it is not giving much options to change or do the manipulations. So there is another software for this, that is Mega. Mega is also widely used software. You can use it. It is called Make. Current version is 10th version, so it is known as Mega X. It allows One thing that Phylogenetic runs on your web browser. Mega is standalone. You can do whenever you want. It doesn't require internet. It can be downloaded for Windows, Macs, and Linux. Okay, it is a free software. You just go to Mega um, Google and type mega. It will mega or this molecular. If you are googling only mega, then sometimes it will lead to different pages. But if you type it mega software, and it will always lead to a correct. You can download from there. It is a very flexible software. You can edit your sequences. You can do much more editing in there. We will shortly doing then one exercise for mega also. You can make tree on the basis of four different algorithms: neighbor joining method, maximum likelihood, your maximum parsimony, and minimum evolution. It also provides option to boost step. Whenever you are going for publications, if you want to get published in good impact factor publications, then you always have to do this bootstrapping. Phylogeny FR, these options were not available. So plus color, shape, size, and layout changes is always there. So always it is a good option for mega. Plus mega is not only just for MSc and T development, it can be used for numerous things. As you can see here, this is a mega startup page. You can align your data, you can get manipulate your data, you can get generated models, you can get distance matrix, you can do all the things here. But today we are specifically doing exercise for phylogenetic tree designing. So we will be sticking to that only. One thing you have to note down that for mega, it is always best that you have stored your data in dot fasta or dot fa format. Okay, how to do that? Simple thing is that you just go to here. Okay, you can open in notepad or word or any word processor. Just like there, you can go to file, just and then go to save as. Okay, once you have moved to save as, you will get this window that you always, whatever software you are using, you will always get this window. From there, always please make sure that save as type is selected as all files. Okay, don't save it dot text, don't save it like any other format dot word. Just shift it to all files, and then after writing whatever you want to sequence, whatever as you want to save, you can write name, then put a dot, and always either type fa or dot fasta. Okay, then click on save. Now your file have been saved in FASTA format. Earlier it was in text format. Your input sequences were in FASTA format. These sequences were in FASTA format, but your file was still as a text. So mega, whenever you are running any text file, you can run, but sometimes it generates error. So it's better you always go ahead and save your file as a FASTA format. Once you are done, then you will go to either open file or directly go to align. Okay, select it, then edit and build alignment. Okay, because we are going for a new alignment, edit or build alignment. Then you will get a different option, that is create new alignment, open or save. If you are working on any alignment session, you can already do that. Or here we are doing for first, then we will retype sequence from a file, and then we will click on OK. It will lead to our input box. You can move to the directory wherever your data was stored. Go to workshop, and then now you have to look for your format. Okay, so we had save in FASTA format. We can select from here FASTA. Okay, then we will open this just like that. Okay, so it is now you can see that your left hand, it contains only your SSN number. It doesn't contains information about your sequence, whatever. If you want that information, you have to do that. You have to make sure that there are no spaces. If space is there, it will discard it. You have to do this and underscore. Always make sure that underscore is there Once you have done, then it will consider it as a single sentence and single word, and it will show there only. Okay, for this exercise we are not going through it, so just we will select all, simply control all, or you can drag and select. Okay, then we will look for W, because we are going for alignment, and alignment algorithm we are using Clustal W. Okay, if you are working with protein, then we'll select this muscle, but in case of um, nucleotide, Clustal W is best, so we will select this, and then align DNA, because we have DNA nucleotide sequence. Okay, it will again ask you to give option you to change any parameters which are default you want to change. It is totally on you, can change it, but you have to make sure that you know whatever values you are putting it corresponding to what thing. So because we are considering that you all are beginner, and or you are not very well aware of these algorithms, so we will not change anything, and we will just simply click it OK. Okay, then what it will do, it will first pairwise alignment, that it will take first sequence, align it with second, then first with third, and so on. First it will do pairwise alignment, then it will go for multiple alignment. As we can see that it is very different from the Phylogenetic or Clustal Omega, whatever we have seen. That's why Mega software is considered more reliable and more accurate for phylogenetic analysis. Okay, once it is done, it will give you something like this. It will not provide any star, or it will not provide anything like that. It will simply give you color coded form. Whatever is conserved sequences, they always come in single color. Whatever is changing, it is always in different. So it will not show any stars here. It is showing on the top of star. It is showing on the top of the sequences here. You can see it is giving you a star, but on the top of the sequence. Okay, once we are get done with the alignment, we will save the file as a mega alignment session. Okay, always keep in mind that we have to save it. If you haven't saved, then it will not you cannot proceed to the next step. So make sure that you save it as a whatever name you want to. For example, I want MSA, so we have MSA and dot MAS. It is that Omega alignment session. Okay, save it. It is briefly for 1 microsecond. You will see a pop-up window, and it will close automatically. Our session has been saved. You can simply cross from here, close from here, or you can minimize it. Then again, we will go to our Mega, and we will shift directly to Phylogenetic, because we are going for Phylogenetic. Then once you click, it will give a number of options that which algorithm you want to use. For example, here I want UPGMA. So I select it, then I will have to navigate to that alignment session we have saved, .mag1. Okay, we select it, open it, and it will give you further option. Now you will see that we were talking about bootstrapping. Okay, so test of phylogenetic, here it is selected as none. You can click on that, and then bootstrap method. You can replicate. Bootstrapping means that it is realigning and reanalyzing and everything. In case of here, if I set 500, then it will realigning, reconsider everything, redo everything for 500 times for better accuracy. Okay, so but we are not going for bootstrapping, because bootstrapping takes time. That every test will get repeated for 500 times. Then submission time type is nucleotide. It is giving us summary that whatever we are selecting. Then we set OK, and you will get this tree. If we have selected bootstrapping, that it will take some time. So it is giving very similar result from here. In case of bootstrapping, it will give a two tree. One is normal tree, second one is bootstrap tree. Now we can change it on our own. As you can see that in Phylogenetic there was not much option. So we can change it, auto fit. Okay, now you can drag and resize it. You can see whatever separation, how much separation you want. In case of branches, how much distance you want, what font it should be used there. You can do everything. plus what is the beauty of Mega is that you can um, put indications if you want, like for example I am saying that this op071 is mine sequencer. Okay, so what I can do, I can do go for this display option, I can select it, then I want to change it, then we go to label. Okay, then we can select a taxon marker, that a shape, certain shape or certain shape or size or color. I can choose, for example I want this op1 is in red. Okay, now second I want a square shape for some other thing in yellow color. Okay, whatever you want, you can select it. Okay, just like that you can change or select it. There are multiple options, multiple color, you can do anything whatever you please with. You select OK, then it will give you an indication here. So it is similarly, you can change color of the font, you can say titanize it, you can make this branch a different color, you can make anything. Then how to save this result? Just you have to go to uh, image and save as whatever format you want. Okay, just simply select the format, or you can copy it and paste it directly from here. So this concludes our Omega. Okay, now because of time constraints that we ran an poll that how long you want this session to be, and we get like 1 hour and so we are slight bit rushing, but I'm trying to cover as much as I can. So we are till now we have been doing for this nucleotide sequence. Now we are shifting to protein, and in protein, in case of protein, motifs and domains are two important things that whenever you are uh, analyzing a protein or you are doing a research on protein, then you always look for motif and domain, because motif and domains are conserved sequences, and they are responsible for the function of or any identification of that protein. Like for example, if you are saying that it is isomerase, so isomerase is how any isomerase, it should be contain a sequences that it is providing it this isomerase activity. So it will be always a conserved sequence. So it is comes under motifs and domains. Motif and domain are two very similar things, but motif is a very small segment of nucleotide or DNA or amino acid. Like 10, 20 amino acid will form your motif. Okay. Example is zinc finger. But domain is a larger and more conserved sequence. So it ranges from 40 to 700 plus, sometimes 700 plus also. But average size is 100, amino acids are there. So as you can see that motifs are a smaller part. Multiple motifs will give rise to domain, and domains will further translated into proteins. Okay, so these are motifs and domains. So popular tools for the motifs and domain is PFAM, Smart, ProSite, Interpro. There are a number of tools. Okay, so to just give you a slight brief on this, we will do this Smart. Smart is for motif prediction. You just copy your protein sequences from database. Similarly, as you downloaded for retrieve sequence from nucleotide, you will get a sequence from your protein also. So for example, here I have already downloaded the ascorbate peroxidase sequence for aryl sativa. So as you can see, it is very stored in very similar format. Just only differences, instead of nucleotide here, amino acids are present. Okay, so you just simply copy it, and you will go to Smart, and this you can paste here, and you can sequence Smart. Okay, so its result, you will get a result in this format. Here you can see this is the, I zoom in a bit, I hope it is visible to you all. See, here is a section is highlighted. This one is our peroxidase region. Okay, so similarly in Interpro, you can Google Interpro. You will get leads to here. You can paste your sequence here similarly, and you will get the result. The difference is there that every software gives a bit different result. If you want to work with domains and motifs, Interpro is a better option because Interpro provides much deeper insights of your protein or domain. Like here, you can see it is giving us a family, it is giving us domain, as well as it is giving us conserved residue wherever whereas conserved residues are present. Plus, it is as well as providing your domain. It also provides you about geoterms. Geoterms are very important whenever you are in research field. That they give you about information about if it is a domain, then what biological process it is involved, what is molecular function, what is cellular components, where it is present. Here it is giving none because maybe its cellular components are not provided or they are not localized to a single cellular component. Okay, in terms of interpro. Similarly, it will provide information about panther geoterms. Here, cellular component is already defined by researchers, so it is showing that this ascorbate peroxidase will always found in chloroplast. It will have peroxidase activity and it will respond to oxidative stress and cellular response to oxidative stress plus H2O2 catabolic process. So from a single domain sequences, a little bit of your protein sequence, it is 251 amino acid sequence, you have solved many things. Like first you will get what is the family, that is it is a thylakoid lumen 19 kDa protein. It's have ascorbate peroxidase, it have peroxidase domain. Okay, like then it will be involved with these biological process. So if your guide or you have to present somewhere and you want to tell that this purpose whatever it's biological process, then you can simply copy paste from here with geoterms, and this result will be considered very authentic. Okay, similarly there is a motif search tool also there. Similarly, you can paste here, you can just select compute and it will give you result about motif, that is a peroxidase domain. But it is giving a very small detail. So I always say that if case of your domains, always interpro scan is always preferred because it is give you much better options. Okay, so with this, we have completed our motif and domain section. Now we will move to structure protein because protein structure prediction is a very important tool whenever we are um, studying about protein. Okay, so similarly, a structure function, yeah, as you all know that if you are learned, if you are from life sciences, we already know that our protein is classified into four structures. That is primary, which is your amino acid residue; secondary structure, that is on the basis of alpha helix and beta sheets, and then tertiary, then it will get fold, and quaternary into multiple folds will correspond a assembly. Okay, real, what is the real word use, that you already know. In case of protein, our drug designing is a very vast industry for protein study, enzyme engineering, disease understanding, all this, because we all know that if there is a structure change in protein, then it can bind with certain enzymes and it can't function properly, which cause diseases. Okay, so protein structure prediction, that is mostly there are three main methods. First is homology modeling, second is ab initio, third is threading. Homology modeling is basic method where what you will do, that it will take your sequence and it just like blast or sorry, this homology, as we see in blast, same function it will do here. It will align the sequence, then known sequence, this structure, it will say that your protein is similar 90% or 95% or 100% is similar to this protein, so its structure will be like this only. Ab initio model is there, that is knowledge based sampling of all possible folds. It, you don't rely on homology. You don't go for just similarity. You are designing and you are folding it on own. Okay, then threading, it's again it is a combination of homology and ab initio with some between lies in between. So protein structure prediction we can do with multiple tools. Again, everything is can be done with multiple tools, but few key tools are sys model you can use, it is web based. Then Google's AlphaFold is there, it is also web based. Then iTesser, Rosette Commons, it is Rosetta Stones to mention. Then these are the key tools. Okay, so we will jump to our again this sequence and just do a quick look that how the protein is you can model. Actually, this takes a lots of time because protein modeling is little bit complex. So again, I have done already done the sequences. So what you will do, you will select start modeling. In case of Swiss model, if you are going to use, then it will take a time and it will give you this homepage to provide your sequences. Okay, always remember again that you have to FASTA format or cluster format. It should be pasted in. Once you paste it's you pasted as sequences. If it is in valid format, then it will convert into a protein sketch with the rainbow color, and then it will provide you different option that you can search for template, you can build model, you can do template alignment or any other thing. But for keeping it basic, we will just directly click on build model. Once you click on this, it will lead you here. Okay, just give me a second. We can just run it. See, it is taking time. Okay, so you can see that from the basic information it have generated a model. It will give you a number of options here, and as you can see here. Okay, so it is providing different options here. So how to check that which one I should consider and which one I should not? Again, as we did in last, what we will see that it is providing us a different here order. GMEQE, you can align any on the basis of any model, you can this align it, and number of this on the basis of this score you can say that whether my protein is correct or not. Okay, so if you select any model, whatever you have finalized, just click on it, and then it will lead to your other things. Okay, it will provide you a Ramachandran plot and all the stats here. Okay, so just wait a few seconds. If its GMEQ value is near 1 or about 1, then it is good. As you can see, Ramachandran plot is here. You see the uh, molecular probability score, it is 1.68. Its class score is 4.24. Class score is your for say that it is steric classes between atoms. Okay, so this class score. This is Ramachandran favored percent, Ramachandran outliers. This is score that your class score should be uh, it must be less than 10. If it is going above 10, then it is not a good fit. If it is less than 10, then it is very good. Okay, Ramachandran favorite should be higher. Your outlier should be less. Okay, similarly for C beta deviation should be less, your bad bond should be less, your bad angle should be less, and you have to keep this discuss with your guide or your mentor that I'm getting these values, then whether should I consider this or not. Because as we know that proteins structure, sometimes we don't get good proteins, but still we want to represent our protein good way, or you want to show a structure, so you can consider a provider with leniency. But always check for these values. Okay. Similarly, then you will see the protein structure. You can rotate in 3D. You can download it uh, from here. Okay. Then there is another protein structure prediction tool that we have already uh, discussed in our starting databases format. That PDB is protein data bank. Okay, RCS protein. From here you can get structure also. So you will select visualize, and then sorry, uh, search. You will select search, and then you will go to this. We want to structure here. We want to structure similarity search. You can select the structure similarity search here. It will give you a input box. Okay, just wait a few seconds. You can also search for protein sequences or anything you can done, just like nucleotide you can do here also. Um, what we are doing, you can search a name by name any term of the protein for protein structure. If you have structure attributes, you can fill in here. From there you can, but we are doing exercise for the homology search. Okay, homology modeling. So what we have sequences. So we will click on sequence similarity, and we will paste here. But um, this particular for PDB, RCSP PDB, you have to keep in mind that you have to need to paste only that sequence. We will not need FASTA format here. We need only sequence. By sequence, I mean from here start to end only. We will ignore this identifier, and because if you paste it, it will not generate a model. Okay, just leave everything like that and click on search. Once you did done, click on search. It will provide you list of models based on the homology. Okay, so you can again apply filter from here, that if you want any particular source organism, you can click on here. If you want taxonomy, on the basis of taxonomy, then you can do on here. You can select experiment model, how much refined resolution you want, and when it 10 numbers of uh, filters are provided here. Okay, so for example, we are going for this only sorghum bicolor, first one, whatever we have got. We open it. Okay, it will provide a basic information, and when it is released, what it's when who released it, who was the author, and all. Plus, one thing you have to see here, that okay, it is still loading. You will get a model here. In case of our this Swiss model, you get a Ramachandran plot here, and then number of values. But if you are beginner, then it can be quite confusing that which value you want to use, which values you should not consider. RCSP protein PDB one is a bit user friendly in this case, that you will get a matrix on the your right hand side, that you can see here. It is a color codec, it is provided, and with percentile ranking and percentile value, that is R free, how much R free is there. Class score, what class score it is you are getting. Ramachandran outliers, how many Ramachandran outliers you are getting. So it is coding, plus it is giving you a visual representation, that if it is your these black threads and black bars are towards your red color, then it is less reliable protein structure. If it is towards blue, that is it's the better and best. Okay, so as we can see, our maximum bars are towards blue. Okay, so that means it is a very good representation of our structure. Whatever structure we are targeting, it should be very similar to this. Okay, once you are done with, you can download file, or you can expand it also like this. Okay, or you can get a 3D structure of it. And your protein ones, PDB ones, will not show you any much detail, but here you can see that it is giving you a good detail, plus option to manipulate it. for this PDB, you get certain options, but they are very limited. Here you can see, sorry, if you scroll bit down, you can see that you can get polymers, you can get water molecules, you can get ligands and plus ions. If you don't want anything, then you want to remove, then you can change from here. Like I don't want to see how where water is present, so I will hide component, it will remove my water. This is the I don't want any ligand stored present, so it will remove ligand. Similarly, you can do anything, and you can export model in any format that whatever is suitable for you. Okay, or you can simply just take a screenshot or download image from here. You download files. Okay, in any format you want, whatever format is suitable for you, you can download from here. Okay, so with this, I conclude today's workshop. If you have any questions, you must have questions. Actually, we can't take individual questions, so we are grouping some common uh, uh, questions here, and I will talk about this. First question was how to analyze your own data, bacterial sequences for whole genome sequencing. Whole genome sequencing is not a basic bioinformatics. Whatever we have um, learned today is is just a basic bioinformatics. For bacterial sequence, if you are talking about whole genome sequences, then
For
you have
the
to,
bacterial sequence, if you are talking about whole genome sequences,
if you are talking about whole genome sequences, then you have to go for genome assembly pipeline and using all that softwares. You have to use those here. We are planning for a workshop for WGS also, but it requires lots of time, so it can't be 1 or 2 hours. It is for days. So if please be connected if you want to do that. Second question is that they have someone have asked that what are tools used in molecular docking. So again, molecular docking for molecular docking, you can say that for molecular docking, you need a molecular structure. So from PDB, you can download the structures, but docking cannot be done with any of this software because for docking you have need a docking software like Discovery Studio or Win Autodock. You can Win Autodock is a free software that you can download it and use it. Uh, Take session in molecular docking. Again, molecular docking is a very wide topic. Plus, it is very complicated. So we can plan out, but we can't. I can't sure that when we can do it. But please, for again, connected. Be connected. We will update it. Someone said that PubChem give me 2D files and PDB format convert. Again, if you are doing for this PubChem, you can directly download 3D format from PDB. You can just copy paste IDs from there and convert it from here. What is this? They can then again question is how do researchers deal with ethicals and privacy issues when working with human genome data? Ethical data, you have to get this approval from research ethics committee, and for as far as privacy is concerned, then actually I can't quite understand what uh, person need here by privacy issue, but as I'm considering it as privacy of your own data. If you are saying that I want to not disclose our data, you can submit and deposit your data, but you can select option that how much section of data or how much data should be available in public domain. Okay, so next is Sefali Senapati have asked, I'm not getting Mega software website. It is not available on website. Just search for Google for Mega software or Mega X. You will it will lead to your page. Just a second, I will just demo it. Okay, so just go to Google. Sorry. Okay, and say I'm looking for Mega X or Mega. So this is software for Mega. Okay, this one will lead to a Mega page. You can directly download from there. Okay, and then someone have asked that. Okay, if you are interested in advanced bioinformatics, then please, as you have already joined our community WhatsApp community, so we will update there, and please give us a feedback so that I can get a better understanding that whatever you want, whatever which sections you want to exclude in next slide, and. I've got a request that some people are asking for mail this PPT and what PPT or they want file also PPT and file. Okay, then PPT files. Everything we will share on we will mail it. Just whatever the you must have a number of Kuldeep. He was coordinating. You just WhatsApp him and that what file you need, and number is if you can write it down, then please write it down. That number is 6393791170. I'm repeating again. 6393791170. Please send request, and you can send your questions also there, and we can we can ask it. Some more questions are there that BLAST can be used to find the exact DNA protein sequence of the gene before editing. Yes, BLAST can be used to get DNA or protein sequence, but given condition is that in BLAST results you have to choose that whether which you want to consider or not on the basis of e values and other parameters. For molecular docking, you need a Linux PC or you need some knowledge of command line. Plus, if you want work with Discovery Studio, then Discovery Studio is paid service. It is a paid software, so you have to contact if you are in if you are a student, then you can contact to your universities or college to arrange that software for you because it is a paid software. And how much time taken to publish after submission of sequence data in NCBI. If your sequence is correct, they will check it. You will get a session number immediately, like in 2 3 hours, or sometimes if they have lots of load, then you will get your session number instantly. But you can choose when you want to publish it. If it is you have selected the option as soon as it is validated, then it would get published within 4 or 5 days because they verify it. So you can't say exactly, but average time is you can consider it like 5 to 7 days. And someone asked that if I have 20 query sequence, yes, you can follow the same procedure for the this phylogenetic tree. It doesn't matter that you have 10 sequences or 20 sequences or 30 sequences. Only difference will be there that it will take more time, and sometimes it will throw that no similar sequence are found. Then you have to manually remove that sequences, but you can do a number of sequences. If you have large data, then I will request you and I will advise you to do Mega instead of Phylogen EFR or Clustal. Okay. Okay, so because of time constraint, we will conclude this session here. And whatever questions I can see that you guys are pasting in live chat, that there are number of still questions which are unanswered. Please WhatsApp them separately in the committee or to pull deep, and I will try to get answer for each and everyone. Plus, still, if you need any uh, suggestions or any help with this, you can always reach to Flamin Lab and contact in person or uh, sorry on online or whatever mode is feasible to do, and we will see what we can do, because it's our duty to help students, especially to students, to achieve their dreams. So, with this, I invite Kuldeep to conclude the session. Okay, so I'm concluding the session. Thank you very much, everyone. Hope this session will be useful to you all, and you guys have gained a lot of data. Okay, so everything with this, I conclude the session, and plus, I request you all that if you you must have that feedback form with you. So please fill that and submit it so that we can improve ourselves. Thank you very much.